vaccines for COVID-19. Just a minute, my slides don't seem to be moving forward. Sorry. Oops. Oh, there we go. Okay, sorry for that. So just so we all are up to speed, just a very brief introduction to SARS-CoV-2, the virus that causes COVID-19. As we all know, it's a coronavirus. It enters the body by attaching to a receptor called ACE2, which is expressed on epithelial cells via its spike protein. The virus then causes the cell to invaginate. The virus is taken in a vesicle, which then fuses with the virus membrane, releasing the viral RNA into the cell cytoplasm. The viral RNA is then translated into virus proteins, virus assembly occurs, and the virus then buds from the surface of the cell. The immune response that is we expect to be developed against SARS-CoV-2 starts when the virus is taken up by antigen-presenting cells, which then present small viral peptides to T helper cells, and these cells then collaborate with B cells to ensure that antibody is made, and cytotoxic T cells, which in theory at least can destroy the virus-infected cell. And as part of this response, we hope and expect that long-lived memory cells will be developed, which will give us immunity in the months and years to come. Sorry, the slide change is being very slow. I don't understand why. Right. So just the first part, the immunology, immunopathology, and potential role of immunotherapy in COVID-19. So this graph is taken from a recent paper in Nature Medicine from a group in China, and shows the proportion of individuals over time who had antibodies to COVID-19 virus. In blue is the IgM seropositivity, in red is the IgG seropositivity, and yellow is the total G or M. And what you can see in this study is that by about 2.5 weeks after the onset of symptoms, everybody had detectable antibodies, the majority of them being IgG. Just as to note, all of these patients had been admitted to hospital, but some of them had relatively mild infections. So that's good news. Everybody seemed to have antibodies. But when we look at the data on which that is based, so each dot here is one individual patient, and the samples were collected over a period of time after the onset of symptoms. And in red here are the IgG responses. And you can see while some people had very high titers of antibodies, some people actually had very low levels. So whilst they might have been above the threshold for description as positive, they were actually quite low in many cases. The same is true for the IgM responses, and there's an enormous spread in the magnitude of the response. Something else that we're seeing over and again now as the papers come out, that on the whole, people who've had severe disease, in the UK that would be people who were admitted to hospital, tend to have higher levels of antibodies than people who've had mild disease. In the UK, those people who were probably not admitted to hospital or certainly not admitted to intensive care. And this is something that's emerging, that antibody levels tend to be higher the more severe your disease, and possibly the higher the level of infection, of infectious virus you had. Nevertheless, it's also true that you can seroconvert to SARS-CoV without showing any signs of infection. And this is just one set of data taken from a pediatric renal dialysis unit, 13 patients, 25 healthcare workers, seen over a period of 3 weeks. These were children coming in several times a week for dialysis. And you can see that only one of the children was ever symptomatic and was PCR positive for the virus, and only one of the healthcare workers was PCR positive. Nevertheless, over a period of 3 weeks, 20% of the patients and up to 40% of the healthcare workers seroconverted despite being asymptomatic. This is just a little bit more detail on what these antibodies are binding to. So this is an antibody binding assay to the spike protein that I mentioned that binds to the cell surface receptor. And these are just titration curves for different individuals. And what you can see is that some individuals have high titers of antibodies that take a long time to dilute out, and others have relatively low levels or antibodies that dilute out very quickly. There is some cross-reactivity with the SARS virus. You can see binding here, but it tends to dilute out very quickly, suggesting it's low concentration or low affinity. And the same thing for binding to MERS. So there is some cross-reactivity, but not if you do a high plasma dilution. And there's no binding, for example, to HIV-1 virus. When we look at neutralization, as you can see here, so this is neutralization of the SARS-CoV-2 virus, and some of these antibodies have very efficient neutralization. They're neutralizing more than 50% of the virus at dilutions of 1 in 1,000, 1 in 10,000. And interestingly, those antibodies that cross-react with SARS and MERS do not neutralize. So the neutralization capacity seems to be very specific for this virus. Very interestingly, this same group have managed to isolate some human monoclonal antibodies from some of their patients with high titer antibodies. And each of these lines is just a different monoclonal they've managed to make, mostly from 2 different patients. These ones in red were deemed to be high affinity, high binding, high concentration. And what you can see here is that these monoclonals actually have very good neutralizing activity. And this is encouraging because if we can make large amounts of these antibodies in vitro, which is entirely possible, they then become potential therapies for treating cases. And that leads me on to this whole idea of convalescent serum or human monoclonal antibody therapy as a treatment for severe COVID-19 infections. So it's a variation on the use of antitoxins. We've used these for many, many years for tetanus, rabies, snake bite, and so on. And they've been used to treat pandemic viruses before. So in the 2009 swine flu pandemic, you may remember this product called ZMapp that was used in the West African Ebola epidemic. Small numbers of people treated, but very positive outcomes. And some use was made of convalescent serum in the SARS and MERS epidemics earlier this century. So the underlying idea is very simple. You have someone who's been infected and recovered from the virus. You take their blood, you check that it contains virus neutralizing antibody, and then you transfuse that serum into someone with severe COVID-19 and hope that they will improve as a result. There isn't much data on this yet. There's been a systematic review that's evaluated 8 studies, totaling 32 patients, and the data are consistent with reduced mortality, increased neutralized antibody titers, and reduction in viral load. But I do need to caution you, because there is a very high risk of bias in all of these studies. They weren't randomized. There was definitely some confounding, and the dosage and duration of treatment varied amongst the studies. On the positive side, there are numerous studies ongoing to evaluate convalescent plasma. At least 22 of those are randomized controlled trials, and there is one trial evaluating hyperimmune immunoglobulin. And these trials are proceeding at pace, and I suspect we'll have some answers quite soon. But antibodies aren't always good. So good antibodies in this case are neutralizing antibodies, but there are also bad and ugly antibodies. So bad antibodies in this context tend to bind to the virus but not neutralize. And then that antibody allows binding of the virus to the antibody receptor, binding receptors on monocytes and macrophages, and that can trigger the internalization of the virus into the cell and can actually increase target cell infection. And that's a phenomenon known as antibody-dependent enhancement of viral entry, and has been seen, for example, in SARS. Even worse, the ugly, is when this internalization process then goes on to trigger excessive inflammation by delivering the virus to the endosome of the monocyte or macrophage, where it triggers a wide range of innate receptors for viruses, leading to the release of pro-inflammatory cytokines. Now, the induction of an immune response requires a little bit of inflammation. So that can be good. But if this goes into overdrive, then it can contribute to the development of a cytokine storm, which is part of the very severe pathology that we sometimes see. So this potential for antibody-dependent enhancement or excessive inflammation is something we need to bear in mind as we start to evaluate the various vaccines that are in development. Another way in which antibodies may play a less than positive role is in the activation of complement. So there is some evidence that complement can play a role in the development of acute respiratory distress syndrome. This has been described in SARS infection, where the activation of complement components leads to the release of a pro-inflammatory cytokine called IL-6 from alveolar macrophages, and that can cause inflammation in microvascular beds across the body. Similar pathology has been seen in late-stage COVID-19, but I'm not aware it's actually been nailed as being complement dependent, but I might have missed something on that. If it is, then we do have potential complement cascade inhibitors that might turn out to be beneficial in such cases. So cytokine release syndrome or cytokine storm is well described for beta coronaviruses. They are known to infect cells of the immune system, including monocytes, macrophages, T cells, and dendritic cells, and induce them to secrete interleukin-6 and other inflammatory cytokines. Cytokine release syndrome is a major cause of morbidity in patients with SARS and MERS, and they have very high concentrations of IL-6, which is a marker of severe outcomes. My understanding is that cytokine release syndrome is seen in patients with COVID-19 and correlates with high levels of IL-6 with respiratory failure, ARDS, and adverse clinical outcomes. And certainly elevated serum C-reactive protein, which is driven by IL-6, has recently been shown to be a biomarker of severe COVID-19 infection. And if that turns out to be a major cause of pathology in a significant group of patients, then we do have some monoclonal IL-6 antagonists, including tocilizumab, which might be suitable therapies. There has been one report of the use of anti-IL-6 monoclonal for the treatment of severe COVID-19. The publication is shown at the bottom of the slide. So in that particular study, 25 patients with a variety of the familiar comorbidities received the anti-IL-6 monoclonal, but they also received other investigational antiviral agents. This wasn't a clean study. Nevertheless, their mean C-reactive protein levels declined very quickly. There was quite rapid radiological improvement. 9 patients at the time of publication had been discharged alive from intensive care. 3 had died, and the others remained in intensive care. And there was a relatively rapid reduction in the amount of time that the patients spent on invasive ventilation. The majority did experience at least one adverse event, but it's not necessarily, it's not the case that could be linked directly to the antibody therapy. So on the basis of this study, anti-IL-6 monoclonal antibody therapy was associated with a dramatic decline in inflammation, with radiological improvement and reduced ventilatory support requirements. But it's again important, this was not a randomized controlled trial, and therefore we need to be very cautious in interpreting it. But randomized controlled trials of anti-IL-6 are, as I understand it, underway. We've also heard in the press in the UK in the last couple of weeks about this hyperinflammatory shock syndrome in children, which has occurred at the same time as the COVID pandemic, and a high index of suspicion that it is linked to COVID-19 transmission. The first set of reports were published from 8 children presenting at the Evelina Children's Hospital in London. They were aged 4 to 14 years, and they had unrelenting fever with variable other symptoms, including rash, edema, and GI symptoms. They all showed the same progression to warm vasoplegic shock requiring supportive therapy and had no significant respiratory involvement. They had variable acidic effusions, elevated CRP, and various other markers of inflammation. Interestingly, they all tested negative for SARS-CoV virus on admission, but all subsequently tested positive for SARS-CoV-2 antibodies. They were treated with intravenous immunoglobulin, antibiotics, and aspirin, and the majority of them were discharged from intensive care within a week. Elevated cardiac enzymes are a feature of this disease in many children, and one child of the 8 died having developed an arrhythmia and a large cerebrovascular infarct. Currently, across the UK, there have been well over 100 cases. I saw a report today that there have been 100 cases in the US, 150 cases now summarised in the US. Here in the UK, the peak seems to have passed about 6 weeks after the peak of virus transmission, again suggesting that there is a link. In Italy, they've seen a 30-fold increase in Kawasaki-like cases since the start of the pandemic. And at the moment, my feeling is the best guess is that this is some form of immune complex mediated vasculitis, where the children have had an asymptomatic SARS-CoV-2 infection, have developed an antibody response, and then have developed some form of immune complex mediated disease. But the good news is that from the UK, at least, the vast majority of these children have done very well and have been discharged from hospital after a relatively short stay. A very interesting paper just published yesterday, and I nearly missed it, a study looking at reinfection. This is the question that people keep asking, can you become reinfected with this virus? So it's a study in rhesus macaques. They were infected, allowed to recover, and then reinfected 35 days later. This is the viral load in the bronchoalveolar lavage fluid during the first infection. You can see that virus peaked quickly and was cleared within 2 weeks. And this is on reinfection, and you can see very little virus in the lungs on reinfection. Each of these lines is a different monkey, and this is just a summary of the data on the right-hand side. There's a bit of a problem with doing this because you're putting virus in, and the question is whether you're just recovering the infectious virus that you put in. So another way to look is to use subgenomic RNA, which is indicative of viral replication. And here we can see in primary challenge, there was viral replication going out for more than a week, but on re-challenge, virus replication stopped within a week and was much lower than it had been in the primary infection. And there was also evidence of a induction of neutralizing antibody after the primary infection. So that's the only data I could find on immunity to reinfection, but at least in macaques, it looks as though there is some. So now on to the question of serological tests. So most of the serological tests that are being trialed are looking for antibodies against the spike protein. So the ones that were causing great consternation in the UK, we thought we'd got millions of them to roll out, and then it turned out we hadn't, are these characteristic lateral flow assays, where you add your sample, which contains possibly some antibodies, it binds with the antigen, it migrates and is captured to identify whether the antibody is IgG or IgM. And typically you get a negative test where the control line is positive. In this case, an IgM positive would give you a line down here, an IgG positive would give you a line here, and both together gives you 2 lines plus the control. In real life, this is what they look like. So this is a study from China. So here we have the control line, a G positive line, and an M positive line. This sample here is negative, although there's a slight hint of an M positive line there. It might be difficult to decide whether that was positive or not. This is a clear negative. This is a weak G positive. This is a stronger G positive, and this is a stronger M positive. In this particular study, the sensitivity and specificity of this particular test was way below the level that we would want before rolling it out, which has to be over 98% for both. So there's a group in Oxford led by Derek Crook that has been comparing a whole range of these different assays, all lateral flow devices, with a gold standard, which is an ELISA assay. So in this ELISA, pre-pandemic control sera that should be negative for antibodies to SARS-CoV-2 were indeed all negative. Samples collected during the acute phase of the illness, on the whole, were positive, although there were a few here that were negative. But all of these individuals were positive by ELISA after 28 days. So that's a nice, strong, good gold standard test. What you can see here, each one of these lines is a different assay. These should all be blue, they should all be negative. So these here are false positives from the test. And all of these should be orange, or certainly all of these should be orange. And what you can see is that there are blue lines here showing that these are false negatives in the test. And as far as I understand at the moment, none of these lateral flow devices have been found to perform at a standard that is acceptable for their rollout in the UK. But there is some better news this week. Two laboratory-based assays. These are variants of an ELISA assay. This first one has been developed by Abbott Laboratories, and in this case, this is the dotted line that shows you whether the test is positive. And just this is on a log scale, so you need to be aware of that. So these are our convalescent samples. The vast majority of them are positive, and these were collected by Public Health England either 14 or 20 days after the onset of symptoms. There are a few negatives here, which are probably early stage, probably 14 days rather than 21. They compared it with sera from some seasonal coronavirus cases, and they were all nicely negative. Some pre-pandemic negative sera, again, all nicely negative. And some sera that would be expected occasionally to give confounding results because of, for example, high levels of non-specific IgG in the serum. And you can see on the whole, this performed very well with very few of the confoundings giving false positives. So overall, PHE found that this had a 99.7% specificity and a sensitivity of about 94%. These were mild cases. Abbott's own data using hospitalized cases found a sensitivity 100%. So that's probably these few that are missing are either early stage or very mild infections. A very similar output from the Roche assay, which I understand PHE have done a deal with Roche today to start using this to screen healthcare workers. Again, the majority of convalescent sera are positive. There are some negative here, and they tended to be early. So we obviously need to wait at least 21 days and possibly 28 days before doing these assays. The confounding serum nicely negative, and the pre-pandemic serum nicely negative. And so, as I said, that assay has been endorsed by PHE, and I understand it's now been taken up by the NHS. So finally, on to vaccine development. At the moment, there are more than 150 vaccines in development around the world. There's a very nice app here run by the London School of Hygiene, which is tracking all of these and updating them. And if you go to that link, you'll be able to see all 150 laid out for you. The vast majority of those are still in preclinical development. There are 7 that have entered phase 1 safety trials, 4 that are in combined phase 1 safety, phase 2 early efficacy trials, or immunogenicity trials, and there's 1 that's that is in phase 2. These 150 are spread across all the different vaccine platforms that are currently being developed. So there are RNA vaccines, DNA vaccines, recombinant proteins, viral vectored vaccines, and then classical inactivated and live attenuated vaccines. And you can see here the vast majority are either viral vectored or protein-based. There are some attenuated vaccines and some nucleic acid vaccines. It's important to realise that with the nucleic acid vaccines, RNA and DNA, there are currently no existing licensed vaccines using this platform. And so I'm not entirely clear what the regulatory pathway for this is going to be. It won't be as straightforward, I would imagine, as for some other vaccine platforms. Viral vectored vaccines, there is one for VSV, and so the regulatory platform for that at least there is a proof of principle for how that would be done. Recombinant proteins, live attenuated and inactivated vaccines are widely used now, and so the regulatory path for those should be relatively straightforward. So just 3 examples of data coming out of those trials, all of them from non-human primate studies in rhesus macaques. The first one is an inactivated vaccine, inactivated with beta-propylactone, alum adjuvated, given by 3 intramuscular vaccinations at 2 different doses. And what you can see here is that you get good antibody titers in terms of ELISA binding. You get neutralization at a titer of about 1 in 100. These are the control monkeys. You get reduction in virus RNA in the throat swab, particularly with the high dose vaccine, less good with the low dose vaccine. And you get reduction in virus in the lung. And importantly, there was protection against pneumonia. So these are the, this is pneumonia in the placebo monkeys. These are the vaccinated monkeys that didn't get pneumonia. I'm not quite sure, it doesn't say in the paper what these were. But the take-home message is it prevented pneumonia and reduced but did not prevent virus shedding. The next one is the one that you've probably heard a lot about. This is the one that's being developed in Oxford. It's a recombinant chimp adenovirus with the spike protein inserted. It's given as a single intramuscular immunization, which is obviously good in terms of the ease of rolling out this vaccine. They vaccinated 6 monkeys, had 3 unvaccinated controls, and challenged them with high-dose virus. Again, it prevented pneumonia. There was no immune-mediated enhancement of infectivity. There was a reduction in clinical signs, that's red compared to the blue here, the control monkeys, and there was a reduction in the amount of virus, and this is particularly the one to look at the amount of active viral replication compared to the controls. But there was neutralizing antibody, but there was definitely virus replication. And lastly, one just published again yesterday in Science. This is the only one that's been through peer review at the moment. The other 2 were preprints. And this is a DNA vaccination in rhesus macaques, 2 intramuscular injections. They reported but didn't detail the clinical symptoms, and they said they were mild. There was no detectable virus replication in 8 of the 25 vaccinated animals. You can see here the reduction in viral load. These are the sham vaccinated, and this is one particular construct which had very good virus suppression both in the lung and in the nose. And they found that vaccine elicited serum neutralizing antibodies seem to be relatively good correlates of reduction in viral load. So if you are intrigued by the vaccination trials and want to get involved, there is an opportunity. NHS Lothian hospitals are set to become a major site for testing of the Oxford vaccine. They're aiming to enrol about 600 frontline health workers, and details of this should be released tomorrow to begin screening on Monday. It'll run out of the Western General in Edinburgh and the Royal Infirmary, and the PI, Dr. Becky Sutherland, is on the call. So if you've got particular questions for Becky, you might want to follow up with her, and you can get information if you want to volunteer at this website here. And with that, I will stop. I want to make 2 acknowledgements. The first is to this fantastic website, immunopedia.org, which is run out of South Africa, and Clive Gray from Cape Town University has been doing an absolutely fantastic job of trawling for all the new papers and summarizing them and putting them on this website. And my other regular source of information is the Science Media Centre, led by Fiona Fox, and her sterling crew who every day are sending out updates on the latest developments on COVID-19. And with that, I will stop, and I'm very happy to answer your questions.
Eleanor, thank you for that.